Good morning, everybody. Welcome to the MPG Primer series. Today, we have the pleasure of having Jordan Rossen speaking. Jordan is a fifth-year PhD student in Alex Price Research Group in the Department of Epidemiology at Harvard School of Public Health. Jordan's current research focuses on methods development for causal variant identification using multi-ancestry GWAS data. Before his PhD, Jordan was an associate computational biologist with the Cancer Data Science Group here at the Broad Institute. And with that, he's talking about population structure and admixture today. Do we have some slides up? Thank you. Feel free to raise your hand and ask questions throughout the talk. There's a microphone in the middle, and I could also bring you one if you raise your hand.
So yeah, I'm Jordan, and I used to be an associate computational biologist here. Now I'd come to this lecture and the other MPG lecture pretty often, and it kind of helped me like figure out what I want to do for my PhD. So it's exciting to come back and give back. And today we'll be talking about population structure and admixture. And so this is the outline for the presentation. We're probably not going to get through it all. Like we'll probably just get through the population structure part. And like, feel free to ask questions because I'd much rather have us get through a little bit of this lecture and have us all actually understand what's going on than get through all the slides. First, we'll talk about population structure, some background, and then go through three different approaches towards inferring population structure. And then if we have time, we'll get to population admixture, talk about an interesting phenomenon that is caused by admixture, and then talk about how to infer local ancestry. And so without further ado, population structure, you can define it in different ways, but it can refer to genetic differences due to geographic ancestry. Another definition is differences in allele frequencies or haplotype frequencies among different groups or populations within a species. And then population admixture, an admixed population is a population with recent ancestry from two or more continents or groups that were previously isolated. So why study population admixture? The first reason is that it helps us understand human migratory patterns and ancient history. The second reason is to improve our power to identify and localize disease genes and understand the mutations that cause disease. I won't really talk about GWAS because that's the subject of the primer for next week, but understanding population structure and kind of handling it appropriately is a prerequisite for doing effective GWAS. Okay, so one of the definitions for population structure talked about allele frequencies. And so to kind of understand what's going on here, we should just start with this object that's called a genotype matrix. Each row of the genotype matrix contains data for an individual that we have mutation calls for. Then each column of the genotype matrix corresponds to a specific mutation that might occur in each person's genome 0, 1, or 2 times. And so this first entry, 0, in the 1, 1 that has a 0 here, that's saying that individual 1 has 0 copies of mutation 1. And then if we go down 1, we can see that individual 2 has 2 copies of mutation 1. And so we're going to see a lot of this genotype matrix today. But if we look at this, we might start to notice something, and it's that some of the rows are colored red and some of the rows are colored blue. But really what we're supposed to see is that like the red rows tend to have a similar pattern of genotypes, and the blue rows tend to have a similar pattern of genotypes. And so just to go into a little bit more detail, we can see it in the first 2 columns. The red rows tend to have 0s and maybe a couple 1s in the first 2 columns, and the blue rows tend to have 2s and maybe a couple of 1s in the first 2 columns. So this corresponds to us saying that mutations 1 and 2 are pretty common in the blue rows, and mutations 1 and 2 are pretty rare in the red rows. And then it switches at mutation 3. And so now red has high values, blue has low values. And so if we want, we can now just directly compute some allele frequencies. And so to do that, we can group all of the red individuals and then sum the number of times that we observe each mutation and then divide it by the number of chromosomes. And so that's 2 times the number of people. So here we're going to sum up this column. We get 1, and then we divide it by 8 because there's 4 times 2 people or chromosomes. And so that's how we get this allele frequency. And that just tells us how common a mutation is in a population. And so what we can see here is that we have these patterns where all the red people seem similar, all the blue people seem similar. One potential explanation for this is that these 2 groups of people have distinct ancestries or have descendants from distinct continents. And that's one thing that could cause this pattern in allele frequencies. And this is exactly what we're talking about when we talk about population structure. And so to make things just kind of more concrete, we can look at a very specific mutation, RS109100034, then look at 3 populations that have been sequenced in this HapMap project. So we have Northern Europeans from Utah, Yoruban individuals from Nigeria, and Han Chinese individuals from Beijing. We can see that this mutation has different frequencies in these 3 populations. There's nothing particularly interesting about this mutation, but it's like an example of how we might have different allele frequencies. But if instead of looking at just one mutation, we choose to look across the genome, we can look at these 3 different populations and kind of quantify the degree of genetic divergence between these three populations. And that's what's happening in this FST metric, where high values mean that 2 populations have diverged, have lots of divergence genetically. Low values mean that there's very little divergence. And so we can do this and kind of do this pairwise analysis across these 3 populations. But so if we want, we can pick any 2 populations and say, what is the genetic divergence between these 2 populations? And maybe that's interesting or not. But it's important to understand that kind of the variation, the genetic variation that exists between populations is much smaller than genetic variation that exists within populations. And so Rosenberg et al. 2002 Science takes a crack at estimating these quantities, and they say that 5 to 7% of worldwide human genetic variation is due to genetic differences between populations. The remaining 93 to 95% of human genetic variation is due to genetic variation within populations. And so given that maybe these population labels are unable to explain most of the genetic variation, why consider this at all? And so in genetic analyses, individuals are often classified based on their continental genetic ancestry. This will refer to the continents or subcontinents from which an individual's distant ancestors have historically resided. And so this is really based on where your genome has come from. And it's distinct from self-identified race or ethnicity. And so, however, this approach of kind of classifying individuals based on their continental genetic ancestry glosses over lots of within continent genetic heterogeneity, but it does capture some extremely important historical migratory patterns. So this model, like, obviously loses lots of detail and isn't objectively correct, but it captures some, it may still be useful. And there exist alternative approaches that allow us to understand genetic structure on a much finer scale. And those are really important. But to continue, let's just look at an example of kind of one approach towards classifying or analyzing population structure. This is the output from a model-based clustering program such as structure. And so the way that we should interpret this plot is that each super skinny colored bar corresponds to a single individual. I can't use this to move, can I? No, okay. Each super skinny colored bar corresponds to a single individual, and then the colors of that bar correspond to the ancestry contribution from different groups. So, for example, in the European individuals, we can see that there's lots of blue. That corresponds to European ancestry. However, we can see that in some group in the middle, there's a sizable amount of orange contribution. So that corresponds to these individuals having a sizable amount of African genetic ancestry in addition to their European genetic ancestry. Yeah. And so, I mean, this is like a very coarse grain approach where we've kind of just said, let's just suppose there are 6 ancestry groups in this population that we're analyzing. However, if we want, we can be much more granular about things. So this is from a recent bioRxiv preprint, Who et al. 2023. And in this paper, they, instead of just classifying individuals based on their continental genetic ancestry, they take the UK Biobank resource and try to classify the individuals based on much smaller regions. They divide the UK and Ireland into 23 different regions. And then in each region, they classify the individuals, or they estimate the genetic ancestry using these very small regions in addition to some other global regions. And so we can see here, this plot is basically just all the individuals that are from Cumbria. And so we can see kind of which color each region is. We have to match the color from the region, from the map to the color in our admixture plot. But here we can see there's a lot of individuals that have primarily Cumbrian ancestors. However, there's a sizable portion of individuals that have primarily Northumberland ancestry, which makes a lot of sense because Northumberland is right next to Cumbria, and we can tell like a similar story about North Yorkshire. And so like that's pretty cool. But the point of this slide is just to show that we don't have to use these super large continental ancestry groups to understand our ancestry. We can be much more granular about things. So that's kind of the background for population structure. feel free to ask any kind of broad questions about our understanding of it. But if there aren't any of those, we're going to go into the inference. So we're going to talk about 3 different approaches for inferring population structure. And so we'll start with these tree-based approaches. These are some of the earliest approaches, and they're kind of, well, we'll see. They're simpler than the other ones. And so what we see here is this is a big tree. And all the leaf nodes at the very end of the tree, the periphery of the tree, correspond to individuals that we have genotyped and are analyzing. And then we can interpret this tree roughly as the tree will group individuals that have similar genetic ancestry or look similar genetically. And we can roughly think of the distance of the path between 2 leaves of this tree. So if I go up and then back down, we can think of the length of that path as some approximation of the genetic similarity or distance between 2 individuals. And so now we'll talk a bit about exactly how we get a tree like this.
So, living with certain region are have mixed ancestry from multiple regions. Does it mean we are referring to a past where there was no mixture, or was there always a mixture? And then they start playing. Because it looks like circular. On the other hand, you say people from their areas. But were they always mixed, or were they not mixed?
Yeah, I mean, it's a very good point. Kind of how we define admixture is a function of kind of how far back we want to look, or what we want to understand is like, yeah, these previously isolated populations. And so I mean, like, we get some information on how we're going to make this call based on populations that have been previously isolated. We might say like, oh, we think of European individuals just been for a long time being genetic. Then we and this is unique European ancestry that has been isolated. So it's a tricky question to answer, but it requires some call. But it's kind of, it's true that if we go back far enough, all populations kind of have to be admixed. But these labels, these kind of continental ancestry groups are still useful for understanding population structure because the populations have been isolated for a long time. And as a result, there has been genetic divergence between them. And so taking that genetic divergence into account is important. Is that a decent answer?
Yeah, thank you, Scott.
Okay, good, cool. Cool. So now we're back to our genotype matrix. And so just to reinterpret this again, this 1 here means that individual 2 has 1 copy of mutation 2. And so here we have genotype matrix for 8 individuals and 10 SNPs, just in our cartoon example. And in order to turn this genotype matrix into some tree structure that tells us about the similarity between individuals, the first step is to compute differences between genotypes. And so kind of the most straightforward approach to doing this is to take one genotype, subtract it from the other genotype, and then take the absolute value of each of those genotype terms. So 0 minus 1 is -1, and the absolute value of it is 1. So we'll do that for every single one of these columns. Then we'll sum up the absolute difference and divide it by the number of markers. And so this just tells us a kind of crude metric of exactly how similar do these genotypes look. And then we'll do this for every pair of individuals. And so we'll end up with, like, because there are 8 individuals in our data set, we'll have like an 8 by 8 distance matrix. And then from here, we just apply some hierarchical clustering algorithm, and kind of like a hand-wavy way, the way to think about these algorithms are either we start with all of our 8 individuals in one big cluster, and then we split it into 2 clusters. And then the way that we decide that partitioning of the individuals into 2 clusters is we try to minimize something like the sum of the distance between the individuals in the 2 clusters. So we kind of have 2 groups that looks similar to each other. Then we'll continue to divide those 2 clusters until everybody has their own cluster. Or alternatively, we'll start with everybody in their own cluster, and then we'll kind of merge these clusters based on the distances to each other. And so I'm kind of not doing a great job like explaining this component of the inference in detail, but I'm doing that because I think the rest of the inference methods are more interesting. And so, but it's definitely, like, if you're interested in this, read more into it. It's cool. But so now, if we do that for a larger data set, than our 8 individuals, we might end up with a tree diagram like this. And so in order to kind of, let's look at this tree diagram for a second. We can see here, it's kind of hard to see here, but oh, maybe I should go back. So like in this picture, we can see the legend for this figure tells us kind of what geographic region these individuals were sampled from, and then they code it based on the shape of these nodes. And so we can see here that mostly we have circles together over here, and mostly we have squares or diamonds together over here. And that corresponds to individuals that were sampled from the same geographic region, having similar genetic ancestries to each other. And so that's what we see here. But there's a few other interesting observations that we can take from this tree structure. The first is that, as I mentioned before, we can think of the distance of the length of the path between 2 individuals as being an approximation of the distance between those individuals. And so what we can see here is that the distance of the length of the path between 2 individuals from the same population is similar to, is only a little bit smaller than the length of the path between 2 individuals from different ancestries or different populations. And so that's kind of illustrated in the cartoon on the right. Like the distance between A to A is only a little bit smaller than the distance from A to B. This is kind of consistent with that slide earlier where I was claiming that continental or population labels are unable to explain most of the human genetic variation. I feel like there's one other thing that I wanted to say about this figure. Oh, and one other thing just to notice is that we aren't able, like the output of this inference method doesn't give us a number of like, what is the % ancestry from this group for this individual. And so we can kind of see it and kind of understand what's going on in the data, but having an explicit estimate of this quantity, which is what portion of your genome comes from a given ancestry group, will be really helpful in some applications. And we don't get it from this type of method. With that motivation, we can move on to a group of methods which we'll refer to as model-based population structure inference. And so one good thing about these methods is that they will give us that number of what portion of an individual's ancestry came from a given group. And so this kind of group of methods is a little bit, I think the name is kind of deceptive because like a lot of these approaches assume some kind of model. But here the model, what we mean by model-based clustering or population structure inference is that we're going to propose an explicit generative model for our genetic data that's kind of based on our understanding of genetics, and then use that genetic model to do our inference. And so here, let's just get ourselves familiar with the data objects that we're going to be working with. So G is our genotype matrix, and that's the same object that we've already interpreted a couple times. GIL is the number of mutations, number of times mutation L occurs in individual I. And then we have these other 2 objects. There's this P matrix. And so the dimension of P is the number of populations that we have contributing to our individuals in G by the number of SNPs. And so each individual or each entry in L tells us how common SNP L is in population K. Sorry, I think I said something wrong there. Each entry in P tells us about how often mutation L occurs in population K. And this is just what we were computing on that first slide where we had the blue and the red individuals. And then we have this Q object. And so the Q object we can think of as our ancestry proportion matrix. And so each entry in Q tells us what the proportion of ancestry K is in individual I. So if Q11 is 1, then we're saying that individual 1's ancestry is all from population 1. And so for now, just to make things simple, we're going to assume that Q is binary and that each row has a single 1 and all the rest of them are zeros. And so this corresponds to the assumption that all of our individuals have ancestry from 1 group each. So there's no admixture. This isn't a good assumption, but we'll relax it in a second. And so just to really drive home exactly what Q is, this is like a sample 23andMe report. And for Jamie, it says that they have 74% European ancestry, 17.5% sub-Saharan African ancestry. And so these are example values that might go in Q. Like Jamie would just be 1 row across this way, and there would be 4 numbers in here. Except it wouldn't fit our current structure because Q has only 1, 1 right now, and all the rest are zeros. Okay, so now we're going to talk about our inference strategy kind of at a very high level. This is an outline for how to do compute a maximum likelihood estimator, which is kind of a general statistical thing that you'll probably run into. But the first step in doing this inference is going to be propose a generative model that relates our observed data. Oh, I forgot to say something. Here, there's a dashed line in the middle. And so we're asking ourselves, what's up with this dashed line? And so the things on the left of the dashed line are the object, or the data that we're assuming that we've observed and have access to. So we can like load G and P directly into memory in our computer and mess around with them. But Q is what we want to find out. This is our unobserved variable that we want to do inference on. And so the first step in our inference strategy is to propose a generative model that relates our observed data to our object of inference. And so in words, this is going to be relate our genotype and allele frequencies to the unobserved ancestry proportions that we want to infer. The second step will be to use the generative model to write out the probability of the observed data given the unobserved data. And so this is going to be, we're going to write out the probability of our observed genotypes given the ancestry proportion matrix Q. And then finally, we're going to find the values of Q that maximize the probability of our observed data. And those will be our favorite values of Q. So this is very not specific, and we'll go into this in more detail. So the first step is to propose a generative model that relates our genetic or observed data to the unobserved data. And so here, our generative model is going to be that the genotype for individual I at mutation L, given that they're in ancestry K, is binomially distributed with parameters P, K, L, and 2. And so to make that kind of less mathy, we can say that if individual I, we're trying to say what is the distribution of mutations for an individual that is in ancestry K. We're basically going to flip a coin twice. And then we'll take the sum of the number of times that that coin comes up heads. And the coin comes up heads with a probability equal to the allele frequency. And so kind of intuitively, each individual has 2 chromosomes. If I tell you that both of those chromosomes come from a given ancestry, then we would expect those chromosomes to have a given mutation based on the allele frequency of that mutation in that population. And so hopefully this first statement is kind of clear now. So the second step is going to be write out the probability of our observed data for individual I. So here this equates to writing out the probability of an individual I's genotype, given that they're from population or that they have ancestry K. And so here, this GI corresponds to one row of that genotype matrix. And we can write out the full probability for GI as the product of each of the probability of the individual mutations. And so we're making some assumption here saying that the mutations are like independently distributed, but we're not going to worry about that for now. But basically, yeah, let's just go back to this. So we're looking at the probability of one row of this G matrix, and we're going to say, what's the probability of GI1? And multiply it by the probability of GI2. Do this for the entire row of this matrix. This gives us the probability of an individual's observed genotype. And so now we'll notice that GIL, given QIK is equal to 1, that's exactly what we have up here. And so we can just go to Wikipedia and look up what is the probability for a binomial distribution. Like what's the equation for the probability of a given outcome from a binomial distribution, and just plug that in for P of GIL given QIK is equal to 1. And so that's exactly what we've done to get to this next line, is we've just replaced this PGIL with the Wikipedia probabilities for a binomial distribution. So now the third step is we just have to ask ourselves, is this quantity maximized when we have individual coming from ancestry 1 or ancestry 2 or ancestry 3? And so now we'll just go into a worked out example of this. We're assuming that we have 2 populations. We're going to try to figure out whether this individual has ancestry from population 1 or population 2. And so the end of the first row of equations is just that exact equation from the last slide. And then we just plugged in the numbers. And so like G11 is this individual's genotype at SNP1. P1L is the population 1 allele frequency at SNP1. Those are all the things we have to plug in here. And so we'll just do that for each SNP. And then we end up with some equation, which we can evaluate numerically, and we get 0.022. So that's the probability that individual 1's genotype would occur if they're in population 1. And then we'll do the exact same thing for population 2. So now we're just plugging in these allele frequencies from population 2 instead of the allele frequencies from population 1. And then we just compute what that number actually is. We get 0.0068. And so we have these two numbers, and we can see that the probability of individual one's genotype is higher if they're in population 2. And so our estimate is that this individual is probably from population 2. Okay, so this approach assumes that each individual belongs to a single ancestry. We just compared the probability of individual one's genotype if they're in population 1 to the probability of individual one's genotype if they're in population 2. However, we know that admixed populations exist and have genetic ancestry from populations that were previously isolated. So can we change our generative model to no longer have this assumption that individuals belong to a single ancestry group? And so here we have the exact same data objects as before. G and P are on the left-hand side, indicating that these are observed objects. Q is on the right-hand side. But now we're going to assume that each row of Q has values between 0 and 1 that sum to 1. I guess there's an extra, whatever. But so it's the same as, I mean, now we're actually in a case where we can give Jamie's 23andMe report, where here now we have contributions from multiple different ancestry groups. And so in our inference kind of at a high level strategy is going to be exactly the same. Propose a generative model that relates our observed data to the object of inference, use the generative model to write the probability of the observed data, and then define the values of Q that maximize the probability of the observed data. And so first, the hard part of this is just going to be kind of transforming our generative model. So before we assumed this distribution for the genotypes of individual L at mutation, or of individual I at mutation L. But then here, this assumes just that QIK is equal to 1, which is saying individuals I only has the K ancestry group. But this isn't going to work anymore. So intuitively, if individual I has 25% ancestry 1 and 75% ancestry 2, then you expect their genotype to look like a weighted average of the allele frequencies from ancestries 1 and 2, with weights 25 for ancestry 1 and 75 for ancestry 2. And so the weighted average of the allele frequencies at SNP-L is just going to be this thing. So we've taken, this is the allele frequency at SNP-L, 25% of that, and then this is the allele frequency at, in population 2, 75% of that. And so we just have to take this number and plug it in for our old allele frequency. That's exactly what we've done here. So here we've just taken the old binomial distribution that we're using before, changed the first parameter to reflect individuals like individual I's personal weighted allele frequency based on their ancestry proportions. And so now our strategy is going to be basically the same. All we have to do is now change our generative model up here that now has individual I's personal allele frequency based on their weighted ancestry proportions. And then we're basically going to do the exact same procedure down here. Write individual I's, the probability of individual I's genotype as the product of all the mutations. And then we'll just plug in our Wikipedia binomial probability equation down here. And so here, this alpha parameter is what we're doing, what we're using to quantify the proportion of individual I's ancestry from population 1. And then 1 minus alpha would just be the proportion of individual I's ancestry from population 2. And then we just have to figure out exactly what value of alpha maximizes this equation for each individual. And so here we have a worked out example. So we're just going to figure out exactly, we can plug in our values for individual I's genotype and the different the different allele frequencies into that equation from before. And now we have an equation, and the only variable here is alpha. And so this is just going to be the probability of individual I's genotype at different admixture proportions. And we can just graph this out. And so here, this is the numerical value for that last equation, where we vary alpha across many values. And we can see that individual I's genotype is much higher. We maximize this quantity when alpha is equal to 0.22, which corresponds to individual I having 22% ancestry from population 1. And so now, up until now, we've assumed that we have access to some allele frequency panel, where we know the allele frequency for each we know the allele frequency for each mutation in each population. That's this P matrix. However, and so this is like, there are many resources that we can use to get this allele frequency reference panel. However, there's maybe that's not so good if we don't really know exactly what populations are contributing to each individual. In our, like maybe there's some population that isn't included in our reference panel that has contributed ancestry to our genotype matrix, G. And so now we're going to try to do this exact same inference problem, but move P over to the right-hand side of the dashed line. And so that means that now, instead of assuming that we have access to some allele frequency reference panel, we're going to infer our allele frequencies from the data. And so now our inference approach is a little bit more complicated because we have to infer both Q, the ancestry proportions, and P, the allele frequencies of each ancestry. And so at a really high level, our approach is going to be just alternate between estimating the allele frequency matrix, assuming that we have the ancestry proportions, and then go on to estimating the ancestry proportions given that we have the allele frequency matrix. And so this is similar to an approach used by software such as structure and admixture, but it's a simplified version of this, but it kind of gives you the intuition behind what's going on. And so to do this approach, we're first going to take in each value of Q, basically just randomly assign ancestry proportions. So say individual 1 is in population 1, individual 2 is in population 3, randomly assign Q. And then we'll use that randomly assigned Q to estimate allele frequencies, given our current estimates of the ancestry proportions, and then proceed to estimating individual ancestry proportions based on the current estimates of allele frequencies. And so like this P, update P given Q step is just taking the means of all of the ancestry of the genotype matrix. What's up?
Yes, that's okay. Thank you. Do you have to provide this approach with the number of populations that you think?
Yeah.
Okay.
And that's tricky. And there's a slide on it in a second.
Oh.
Yeah, great question. Yeah, I mean, here in all of these matrices, you can see that there are at least 200. We're indexing from 1 to K. So the question is, how do we specify K? And that's a tricky question. Okay, so here we just take turns estimating the allele frequencies given the ancestry proportions, then estimating the ancestry proportions given the allele frequencies. This is estimating the allele frequencies, just taking means of our genotype matrix, and then updating estimating the ancestry proportions given our allele frequencies is exactly the procedure that we went over like 3 slides ago. So we're going to do like this thing a bunch of times until we reach some stable solution. And it's kind of wacky that we can like find the true ancestries just starting from random assignments, because like initially this allele frequency matrix isn't telling us very much, but it works out, which is cool. So yeah, as you did that, do we have to provide this method with the overall number of ancestries for sort of population? And the answer is yes. So here we run this algorithm on a big data set, varying numbers of populations. And we can see that when we set the number of populations equal to 2, the method just basically spits out ancestry proportions that correspond to African plus European ancestry versus Asian plus American plus Oceanic ancestry. That's kind of the main division that we get from that. But then as we increase K all the way to 6, we start to get much more granular ancestry definition. And then like when we hit 6, we get this smaller group of individuals that have their own kind of ancestry category. And so it's not at all obvious which of these answers is actually right. And that's a major disadvantage of this kind of method. So maybe if we have some ground truth about how many ancestries we expect to use them to see in the data, this will be good. But it's tricky. It's not obvious what the correct number of K is here. So that's a good motivation for moving on to PCA-based population structure inference. So principal component analysis is a very general data analysis technique. You'll see it in all kinds of fields. We kind of just try to get the intuition behind what's going on. Suppose we have a data set with 2 features, X1 and X2. The first principal component is going to be the feature along which the data varies the most. Or we'll kind of try to consider all the potential axes in our data, and then find the one that spreads the data out the most along this axis. And so then once we can compute the principal component one, we'll proceed to identify the axis which explains the maximum amount of variation, kind of taking into account the first principal component. And so here, I mean, in this example with two features, it's kind of silly, kind of like there's only one place that's having principal components together. But we can do this with much larger data sets. So here, this is what principal component analysis will look like when we do it on genotype matrix. So we have I individuals and L mutations. And then basically our first principal component will try to capture as much of the variation in G as possible. And so basically what the first principal component will capture corresponding to this, well, this first row in this matrix, there's basically going to be many mutations that co-vary. We'll kind of try to find the maximal combination of mutations that co-vary together. And if we think about it, like what group of mutations are very likely to co-vary together? A reasonable guess is something like continental ancestry, because if individuals have ancestry from group one, they're going to have a bunch of mutations that aren't at high frequency in mutation in population two. And then similarly, individuals from population two will have a bunch of mutations that aren't seen in population one. So we're going to get lots of shared mutations that are varying together, and that might be what you expect. And unfortunately, it's exactly what you get. And so this is PCA run on one of the really early population sequencing data sets called HapMap. We have European, Nigerian, Chinese, and Japanese ancestry individuals, and we can see that when we do PCA on it, PC1 primarily captures the difference between the African, or the Nigerian and the non-Nigerian individuals, and then PC2 primarily captures the difference between the European and the non-European individuals, indicating kind of these axes of population structure maximally capture lots of variation in the genotype matrix. And so interestingly, principal component analysis can also capture continental admixture. And so here, in this figure, the authors have concatenated Native American, European, and African ancestry data set with Hispanic with genotype from Hispanic and Latino individuals, as well as African American individuals. And so what we can see here is that many of these populations kind of occupy these distinct regions of this plot. And we'll also see that but there's also lots of variation within these groups. And so individuals that identify as Dominican, from being from the Dominican Republic, have kind of this unique admixture profile. So we can interpret the intermediate values between these far groups as these individuals having contributions from different continental ancestries. And so here it's probably like, it's important to say a lot of this admixture, like for example, in the African-American individuals or between the African and the European individuals, this reflects some legacy of slavery that appears in our genetic data. And so it's probably good to not just treat this with complete detachment and kind of acknowledge the historical events that have led to this. So the next figure is, it's like everyone's favorite figure. And so it's from November et al., 2008, Nature. And what the authors do is they take a bunch of different European ancestry individuals and do PCA on it. And on the left, they have PCA. So this is just basically the maximal axes of variation in the genotype matrix. And if you compare the figure on the left to a real map of Europe on the right, they look kind of similar, right? So it's like Portugal and Spain are like down here, like it is on a real map. And we have like Great Britain and Scotland up here, like in a real map. And we go to the east and like there's some France. And so like, how crazy. It's like, we just had the genotype matrix and we remade a map of where these individuals are from based on it. So this like really, these last two examples have really shown us some of the advantages of principal component analysis, where it's like, here we're able to like get estimates for admixture proportions. I guess we could get that from the model-based framework too. But here, we, rather than having like super distinct isolated populations, we have kind of allele frequencies that vary with a gradient over space. And so it's like, there's probably small levels of migration a long time ago between people that live in France and Spain, but not a lot. And as a result, we kind of have these continuous clines of allele frequency differences. And so PCA, it would be really hard to decide like what are the correct populations, or what's the right value of K to use here. But PCA makes it easier so that we don't have to think about that so much. We're able to capture these continuous differences in allele frequency across geographic space. And so a pretty important question. Oh, there's a chat. Is this okay? Okay. So a relevant question here is model-based ancestry inference or PCA better? And so model-based inference approaches are motivated by explicit genetic models with distinct ancestry groups. And as a result, they impose corresponding constraints. So it's like the ancestry portions are guaranteed to sum to 1. They can't be greater than 0 or less than 0 or greater than 1. And the method just immediately spits out exact estimates of the quantities that we're interested in. So that's an advantage for model-based inference approaches. And they also allow us to impose like some prior knowledge on the data. Like if we know that there are 3 source populations, we want to think about it that way, we can impose that understanding of the data on our inference. However, PCA may better capture situations where population structure varies continuously. If we don't want to specify that number of populations, then we'll just compute a bunch of PCs and kind of see like where they stop capturing things that look like population structure. Yeah, so those are advantages of PCA over model-based inference, but really both are useful. And if you're doing this kind of research, you will find instances where you want to use each of them. There's no, these chats, do I need to worry? Cool, no. So how much time do I have left?
That is a great question. I think we are, it would be wonderful to have some Q&A. We're about 4 minutes before we have to do the changeover.
Cool.
And we're kind of at a natural stopping point.
Definitely. Let me do one last, like.
Yes.
Well, there's like this slide. And so.
This is going to be my question to you.
Great. So I think if you want, if like this is interesting and you want to learn more about MPG, like when I was a computational biologist, I didn't really know anything about this, and I got excited about it by coming to these. The first is you should keep coming to the MPG Primer and the MPG seminar. The next is that my PI teaches this EPI 511 class at HSPH in the spring, and he'll let you probably let you just audit it if you don't can actually take it for credit. And I think it's an awesome class. You go over like lots of stuff like this in more detail.
And he also shares his slides online freely, right?
Yeah. And so like a lot of the slides from his class I stole for this, and they're all free online. So you can go check those out. And like I use those as a resource all the time. And then this book is pretty new and like not actually finished, but there's like the first 3 chapters from it. And a lot of the slides I also stole from that. And this book is awesome too. So check it out.
Stole with appropriate
yeah, with credit.
crediting.
But yeah, are there any questions about the subject matter or presentation?
Let's first give a round of applause for Jordan.
Thanks, Jordan. That was such a good primer. I was wondering, do people like combine PCA and model-based? So if you say, if you want to get the ancestry proportions, could you look at the PCA, this like and identify how many Ks you want to specify and then put that as?
It seems like it's more do PCA, pick those components that seem real, and then just use that as K. I know in some of the model-based clustering literature, there is discussion of how to choose K. And it's possible that this is suggested as an approach, but I don't know if it is. It's a cool idea. I like it.
Thanks. Nice.
I've got a question while other people might want to come up to the mic. I had a question about the assumption about assuming independence between SNPs for the generative models. And given that we know that assumption is not true, and do these models still perform well, or when do they perform not as well? Yeah, so it's true that it's typically, if we just kind of use every SNP in the genome, oftentimes SNPs 1 and 2 will be in linkage disequilibrium, which means they co-occur or never co-occur. And that violates our methodological or our generative data assumption that G11 is independent from G12. And so typically, I think the advice is to just
prune your genotype data such that you don't have adjacent SNPs in LD or close, nearby SNPs in LD. But I bet that kind of to what extent you do that pruning, how strict about it you are, and you can probably get away with not being perfect about things and it'll still come out mostly right, because kind of the dominating factors in your genotype matrix will be these ancestry proportions, not this kind of small region of LD. But you probably get better estimates if you prune your SNPs better. And I think that some of these methods also do relax this assumption that there is independence between adjacent SNPs, but I'm actually not that sure about that. The approach might just be you have to do some pruning based on linkage disequilibrium.
That sounds great. We're at time, but we don't have to move today. The seminar will be in here, so you guys can get your breakfast. And then let's just thank Jordan one more time for his wonderful primer.